Yeah, or watching as part of the live stream. Who knows? I mean, C-DEBI as a YouTube channel only has 6 subscribers, so I don't know who would have gotten a notification about that.
Everyone's sharing it with their friends on Facebook.
Maybe that's what it is. It's so hilarious. Look at this person who's being comically just, never. Anyway.
I did see your face as well, because it's
they
like
do see the face. They see the face.
Close up too. So like, yeah.
Welcome YouTube. Okay, so we can get started. I don't need this up right now. All right. Welcome back from lunch, everybody. Everybody have a good break. Actually, I'm gonna grab my. Again, welcome to YouTube. So the first session that we're gonna do this afternoon is gonna be involved around quality control of Illumina sequences. And so you might be like, well, that's a weird thing to start with. Like, I'm kind of here for the actual interpretation of data part of the bioinformatics. When do we get to that? And all I have to say to you is it's really important. And it's kind of important. With the data control side of the quality control, what you get off of a sequencer, which is really what we're all really aiming for at this point in time. Mostly this workshop deals with DNA, right? But it's the same if you're doing some type of transcriptomic analysis or metagenomics analysis, and true for all other methods. But you have to do some version of quality control. And yes, there are lots of services out there, right? Out there right now who will do sequencing for you. They will send it to you in this fun internet packaged output for you where you can look at things, and they will tell you how many sequences you get, and they'll tell you and promise you that they've quality controlled it and everything's fine, and just use the data that they give you, everything's great. And all I can say is even in those situations, there can be holes or gaps in what those companies are doing for you, and the data that you might be getting might be less than the ideal that you can aim for and what we're doing, or for data that you want an output. And so this is an example that we're going to work through. We're going to work through some actual raw Illumina sequence data. It's a little old at this point, right? So even now it might be at this point you maybe not even get something that's as low quality as what we're working with. But we're going to work through that, and the process that we're doing is actually very much favors really high quality sequences. So we will actually throw out quite a bit of the data that we're working with to get at the really core high quality stuff. This is really conservative. Nowadays, a lot of the stuff that we're going to work with tomorrow, especially with the new lines of assemblers that are out there, new versions of assemblers in those cases, they actually don't have as many pitfalls. You don't have to be as cautious. So you can actually use some variation of this where instead of being as conservative as we are, you can be half as conservative and still get some very good data that will be used very effectively by the assemblers. It won't cause any problems or make have any large issues. But this is just one example of what we're going to do. Quality control is a really good example of something where there are hundreds of different ways of doing this. And so it's, these are some tools that have been around for a little bit, so they're a little bit more tried and true. If you're looking for things that are a little faster, newer, shinier, there's a whole bunch of them. You can go and you can look for those, and they will do different things depending on what your question is and what you're trying to get at. So that's just a little input to what we're going to be doing here. And so what are we going to do for this? So what we're going to do is so if we're starting, I'm all the way back here at the base level of the virtual machine. If you move into the EcoGeo folder and look here, there's one called file quality control. I'm looking at the wrong thing. And you can CD into quality control. And if you hit LS, you can see there's a number of different files hiding in there already. And so we can look at what a output looks like. So if in the first session we were working with in the introduction, we were looking at FASTA sequences, which are file structures that have the header line and then some information that's genomic in nature, either genetic or transcriptomic, or in some cases proteins, right? Where you just have a header and sequences. The files that we're going to work with here with the quality information are FASTQ files, the Q being quality in this situation. And so if we take a look at what these files look like, imgr1.fastq, you can see they're already very different than a FASTA file. So the header caret has been replaced by an @ symbol. Still kind of the same idea. This is your identification line here. So this is your sequence ID. And then this is some information that goes along with it. And then the length that's being stored here. This is the sequence itself right here, highlighted here. Then you see this plus sign, and then there's a whole bunch of other letters. And so these other letters are actually the quality score information. And so when we get into that, we can actually visualize that as numbers. You can get an idea of what good quality and bad quality looks like. But this is information that's coming off the sequencer itself, and it's telling, we have stored in this file information about how good the signal was for any one particular base on the sequencer. And if you scroll, not that way, but if you scroll down, you can see there's all sorts of, like, here's a caret here representing a different score value. Here's a whole bunch of @ symbols, which makes using the @ as your header pullout pretty problematic. We get around that in a little bit. Normally the way you do that is by targeting some longer string of your identifier, right? So if you were to try and grab out all of the identifiers from this sequence or this file, you could do that by doing the grep command that we were doing earlier, but then instead of just using @ you'd use @srr, right? And that would pull out everything that had that starting information there, and you would avoid lines that actually have ats in there representing the quality of the sequence. And this goes on, and it's shown to you here in the slide as well. Oh, and then just to remind everybody, I mentioned this earlier, gray boxes are boxes that we will do. Those are commands that you can execute. If the box is black, that's something that has either is not something that we're going to do, or it's something that took a long time to run, and the file that it generates is actually somewhere else hidden and ready to be used for everybody in the room. Okay. All right. And so the first thing that we're going to do is that we want to have some way of visualizing the data that we're going to work with. Because looking at a bunch of letters and trying to understand how good the quality is can be a little problematic. And so installed on the virtual machine is a user interface called FastQC. And so if you type in FastQC and hit enter, this box should appear. And what we're going to do is we're going to go to File, Open. You're going to have to dig into this a second. So we're going to go File, Open. It should open up a box like this. We're going to go to EcoGeo. We're going to go down to Quality Control. And then we're going to pick the one that says 10000002065R1. So it should be this first one. And so this is a subset of a larger data set. So the way the FastQC works is it looks and randomly subsamples sections of your data at 10,000 reads increments. But if you are going to put in 10 million reads, it takes a long time to read. So we're just going to put in 100,000 and allow it to process really quickly. And so that's going to go up real fast, hopefully. And then you get some information that looks like this. So we're going to go over some of this output so that we can get an idea of what's going on. Just making sure. Yep. So this is what we're going to do. So the first thing that we're going to look at here is this per base quality, sequence quality. And so this is what I mean by needing to go through and actually quality control your sequences. So what you have here are 3 different regions represented by red, yellow, and green that represent the kind of buckets that people put good sequence values into. So sequences below, a sequence quality score below 20 is normally not considered that good. In this range between 20 and 28, it's okay. And then between 28 and 40 are kind of what we consider like really high quality scores. And so you can see, and this is very common with Illumina sequencing, and even back in the day with Sanger sequencing, you end up with this essentially curve that happens where as the length of the sequence increases, the quality of the bases decrease on average. And so you can see these box plots and this moving trend line that give you this output where it starts off really high with the average here for, the first, this is the first 30 bases or so, is really high quality, 39 or higher in terms of quality score, and then it drops off. And so even though our average stays pretty high, we have this like very wide range of sequences, some of them which are the lowest possible quality that you can get, which just means there's nothing there useful right there. So we don't want that in our data set. We don't want that as part of our initial information, and we're going to get rid of that and try and clean this up really well. There's a lot of other information that you can see on this part of the bar right here. So this is a lot of more information that you can get from these analyses. And this gives you a little bit more hints on some of the other stuff that you can run into in problems when you're quality controlling. Double check which one. So the one I want to highlight is this one here. So this looks like a problem, right? So this has got an X here saying that we've registered this as an issue. But I'm going to tell you right now, it's not an issue. And so this is a result of the fact that FastQC is a program that's designed to work with sequences from human sequencing projects, where the GC content has this very nicely mean distributed curve to it. These are environmental samples from microorganisms these are from the deep subsurface. And you can see there are two peaks. And there's a mean GC peak here, and then there's like a high GC peak here. That high GC peak is actually high GC organisms. Those aren't, they don't adhere to the normal mean distribution of what the program's expecting. So it's calling an error, but at the same time, it's actually not an error. So that's useful to know. And then there's a whole bunch of other things here that have all been given check marks. Right, so this is, you can look here, this will change after we make some adjustments, but right now all of our sequences are 100 base pairs in length. And so if you looked at anything below 100, there's none. If you look at anything above 100, there's none. So all of them are perfectly right here in one spot.
So for this one in particular, for stuff I work with, there's tons of indels. So this one would probably be all over the place.
For the sequence length distribution? For the sequence length distribution,
with that.
So when you do Illumina sequencing, right, you're fairly limited, right? This raw stuff that they give you will be exactly what the number of cycles that you've told it to, right? So if it's a 1 by 50 base pair, 2 by 50 base pair paired end, it's going to go 50 cycles. You're going to get something that if you start with the raw data, it's going to be 50 base pairs in length. Some of it will be good quality, some of it will be bad quality.
Paired end 300 base pairs because there is a variation.
But that will still mean that the sequences that you get back, right? So the 300 base pair variation that you're talking about, the variation will happen in the library construction part, right? Where you can have something where you say you want to target a 300 base pair region, and there's some variability in that size. What you actually get off the sequencer is fixed at the length that it can be. And that's what you get. And that's what the quality comes in, right? Because not all of those base pairs will be correct. And that's why we're going to do some quality control to it. But the actual data that you get off the sequencer is fixed length, because the computer and the machine that's doing it doesn't actually know when to stop and when not to stop. It just knows that it's going to run 300 cycles, and it's going to give you the data that has that 300 cycles, and it's up to you to do something with. But you're absolutely right. In the library prep stage, you can have a range of insert sizes that go into those libraries, and that will depend on the sequences that you're putting in. But the actual data that you get back out is fixed at length, the fixed length that you can get.
It wasn't for the stuff I did.
Well, then you might have gotten something back from the sequencing center that had already done some quality control for you, right?
Then maybe they did.
Yeah.
There was variability because we were looking.
Yeah. This gives you some information about how many of the sequences are duplicated or not. So right now we're in a really good setting, but so this would be something that if you were doing a 16S amplicon run, this would look fundamentally different here than it does here, because from a metagenomic sequence, you're kind of expecting every sequence to be different. There is some amount down here. You can see 98.7% are fine, but there's some small portion of them that actually have duplicates, right? So those can be library construction errors, issues like that. Again, if this was an amplicon sequencing run with 16S, you'd have all sorts of variability here, because you're expecting to see maybe identical sequences depending on how diverse your system is. And then the last couple ones here, which are not going to show representation because The data set is fairly small. So this is where you would find overrepresented sequences. This is where you start to find indications of, are your primers still part of your sequences, right? So this would be a segment of DNA that was used to prime the sequence in the sequencer or in your library construction step. Maybe it's a multiplexing ID that hasn't been trimmed. And this would show up as an overrepresented sequence, because naturally you wouldn't expect 1,000 copies or 10,000 copies of the exact same fragment over and over again. But this is a region that you can hone in on and be like, we don't need this anymore. Let's find a way of removing that. So that's what that's there's for. So we can do the same quality assessment, but now we can do it for the reverse read. So that's listed instead of R1, that's R2. So we have the reverse read going in and or forward read going in and reverse read coming out. You can hit OK, and that will run. Boop, 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 boop. And again, we can take a look. You can see this is even worse, right? So if we were to compare these sequences to these sequences, this has a much lower average, starting much earlier in the output of the sequences, which is going to be shown here. It's going to be shown here. And so again, so this is not registering as failed, but you can see, I think this is an example. Yeah, so this is an example of a failed or a non-useful duplication amount, right? So this is an example. This was an example from a real data set that I was working with from sediments, where they didn't get enough of their target DNA out of there. And so there were these artifacts that were being generated during library construction, where if you looked at it, you could see that most of their data was actually happening in this region here that had a lot of sequence duplication happening to it. And so this plot shows you, the blue line shows you what your data looks like. And then the red line shows you what happens if you remove that duplication. So if you remove it, it all of a sudden, most of their data disappears if you make it down to just one copy. And so a lot of the data ends up being thrown out in that situation. It was not a good sequencing run. And then the last example is a slightly larger file so that we can see some of these other flags. So that's this is 250 or 2.25 million, 2.5 million reads in the same file, but subset differently of the forward reads. So this might take a little bit longer to go up. You can see it counting very slowly. And so you can imagine if you're doing 100 million reads or a billion reads, this is going to take a little bit of time to do. But it is useful sometimes to visualize them. So again, so if we compare this quality scores, you can see, subsampling a slightly larger amount really doesn't change our averages. But you can see now we have this K-mer content error. And so this is, again, so these are the first 9 base pairs that we're looking at, base pairs 1 through 9. You have these kind of peaks. And so this probably is an indication, now that we're looking at a larger data set or a subset of our data, indication that there's some carryover in primer sequences. There's some carryover in the sequencing adapters that we're going to have to remove to get to the true data, right? Because it's great that they all have it. That's super useful in the sense that, like, hey, we can check to make sure everything worked, but it's not great because there's no real data there, right? So if we did analysis using those first 9 base pairs, we're not going to get anything worthwhile out of that because there's no biological meaning there. So let's forward through these. So let random priming can show these K-mer biases. All right, so what we're going to do for this step is we're going to remove the residual primer sequences, and then we're going to try and increase the overall quality scores by removing poor quality sequences. And so we're going to do this in 2 different methods, just to give you an example. One, we'll take both of those steps and do it at one time using a program called Trimmomatic. The other version of that uses 2 programs split up, right? So it will use CutAdapt, which cuts adapters off of your sequences and removes those residual primer sequences. And then Sickle, which then does the quality control step. And so the idea is that we can compare and contrast these 2 different versions of quality control, and we can look at the fact that they hopefully, if they're doing it right, will successfully find remove and get to the same core high quality sequences that we can then work with downstream. So when we're done with FastQ for right now, so we're going to hit the X to close out so that we can go back to typing on the command line. Okay, so a little background on Trimmomatic. So Trimmomatic is a Java-based program. There's a hot link to the manual. Again, this should also be in the install list that everybody has access to. And so some of the features about this is that it comes with a file that has common Illumina adapter sequences. So that's one of the steps that we can use by comparing the sequences to a known list of adapters, and we can use those specifically to remove them. And then it's also multifaceted in the sense that we can ask it to trim adapters, trim by quality, trim by specific regions, and is really useful because it can maintain paired end reads, right? So instead of processing each set of reads separately, it's going to process them together, and we'll keep that information stored about where they are instead of having a sequence that where we get to. So what we'll do is we'll actually probably set a length cutoff to make sure that we're only getting high quality sequences at some point. And instead of finding a sequence that it just has no good information and throwing it out and losing it and ruining our paired end nature between the two files, what we'll do is we'll actually maintain essentially a zero length sequence so that future programs can keep track of matches that don't work anymore. And you'll see how that works in a second if it doesn't quite make sense right now. Okay, so let's start typing. Look at all the typing we have to do. So you have the choice here. I can make this full screen and everybody can look at it, or I can type it along with you, and then eventually it'll be visible because the virtual or the command line's a little bit larger. Does anybody have a preference? I'm typing. Thanks, Katie. All right, so there's a lot here. And so we're just going to go take it very slowly. This is one of those examples where after you've typed it the first time and you've gotten it to work, when we get to the next slide, copy and paste is your best friend. Don't worry about it so much. And just as a note, it works really well where if you've gotten something that works and you're working on your own data and you've got stuff that's going on, keeping an actual running list of the stuff that you're working with in a separate text file, doing your own copy and paste, moving something out, moving something in. This does two things. One, it means you don't have to spend the rest of your life remembering what the correct order for Trimmomatic is and all the flags that you need to have to make sure that it runs properly, which is great because who has space in their brains for that? The other thing is that it allows you to have something that you can then actually work with manually, copy and paste, and change individual files in a format that isn't the command line where you might be having to go back and forth. You might be having to hit up and down and using the marker key to go back and forth like this. You can actually just highlight something, delete it, put something else new in there. All right, so let's do this. So we're going to call this, because this is a Java-based program, we actually have to call Java. So we're going to call Java until it's running a program called using the -jar. The program, we then have to tell exactly where the program is that we're going to be using. And so the program is hiding in home, C-DEBI, Bioinf programs, Trimmomatic. All right, so I use tab there to get that a little faster. I'll go a little slower here. So it's home, C-DEBI, Bioinf programs, Trimmomatic with a capital T, and then lowercase Trimmomatic, and it ends with a .jar, which might be easier to see on your computer than mine because it might end up, not wrap over the lines. Okay. So next thing we're going to tell it is that these are paired end sequences with a capital PE. We're going to do a space dash Fred33. And so this is, we're telling the program what type of quality scores we're going to be using. So Trimmomatic was released in a time where there were lots of other sequencers that people were competing, Ion Turret and all sorts of things like that, which actually use different quality score values. And so Fred33 is the standard values that were used in Sanger sequencing and are used in Illumina sequencing. If you were going to be using some Ion Torrent or 454 sequences, which I don't think people use anymore, but if you were to be using those, you would have to use a different flag here to use a different quality score metric. All right, and so then the name of our first file is IGM. If we put a 0 and then hit tab, it goes all the way to the R. can hit r1.fastq. So that's our first file. And then our second file will be R2, tabbed all the way out. And then the next set of line or information that we're going to put in are actually the output files that are going to be generated. And so right now they're in a shorthand, just so that this is a little easier to keep track of. It looks like a lot of letters and numbers, but trust me, it's a little easier. But if you want, when you actually do this yourself and you have a final product that you're happy with, you actually want to make sure that you give these real names, that you call it IGM 000 trimmed or something like that. Something that gives you some indication that that you've already quality checked this information. So what we're going to do is we're going to say R1 underscore PE, R2 underscore PE. And so these are going to be our results, paired end, that have both sequences in the pair have passed quality control. And then we're also then going to dump out, oh, actually, let me do that one more time. So it's R1 underscore PE, R1 underscore SE, sorry, with a space. And then R2 underscore SE. So it should be PE, SE, PE, SE. Again, the PE ones are the paired end ones that both passed quality control, kept in the same order. SE are single end. So these are ones where one member of the pair had high quality and were kept, and then the other one was thrown out. So you have two different files there for that, right? So R1 single end are all the R1 files that had good quality, but their mate didn't. And then R2 is the opposite of that. All the good quality ones in R2 that didn't have a good mate in R1. All right, we're getting there, we're getting there. So now in all capitals, we're gonna type in Illumina clip. And so we're gonna tell it to trim the adapters off here using the sequences or the ones provided by Trimmatic. And so this is going to be another address that we're going to not type in. Yes, not type in. We have to type in by hand. Home, C-DEBI, Bioinf programs, Trimmatic -035 -adapters /true seek 3-p-fa. So now we've given it a location to look for a FASTA file that will find all of the adapters that are in the sequences remaining. So this gives a handful of adapters to look for. And then we're going to actually tell it some information about what it needs to consider to match. And so that's what these numbers are. So you're going to do :2, :30, :10. And this gives you some information about you're looking for 10 base pair fragment where some number in a sliding window have to be correct for it to actually consider it an adapter. All right, next command or next part is sliding window, which we're going to tell it now. We want you to, Trimmatic, we would like you to go over every 10 base pairs and take an average. If the average is 28 or higher, keep the information. If it drops below 28, that's where we want you to say this is no longer a good sequence. So that's what sliding window does. And then we finally tell it, if you go through all of this and you find that your sliding window cuts before the 50th base pair, throw it out. We no longer want that information. So if I've typed this out all correctly and I hit enter, this should run. We will find out in a second. Completed successfully. Great. I knew that was going to happen. I guarantee I knew that. And so you get some information here.
Can you repeat what minlength was?
Minlength, yeah. So minlength is saying it's going to look for across all the sequences. If the point where it's decided this is where bad quality is, okay, gives you a length less than 50 base pairs, okay, it's just going to throw out that sequence.
That per sequence or one merge?
Per sequence. That's how you end up with the paired end, single end type of stuff. All right, and so for the output here, so we looked at 100,000 sequences. 68% of them both survived, and then another 21% were just the surviving forward reads, 2% were just the surviving reverse reads, and then about 7% actually both sequences of the pair were below this quality threshold that we've set and have been thrown out. And so you can play around with this, right? You can lower the min length and say, okay, maybe everything above 40 base pairs is fine. You can change that quality score from 28 to 22, right? Which moves it from that line between the yellow and the green to between the red and the yellow. And I've seen papers where they do lots of different variations of this. It really is about managing your data and what you're trying to look for. And so if we look at in shock and horror at this, oh, this is more just on the output, so we're not copying that again. Whew. Right, okay, so this is explaining the outputs and the inputs here, how the Illumina clip is working. So this is a summary, so this is the information down here. So this is looking, so this breakdown of 2, 30, 10, what does it mean? So this is the maximum mismatch value, right? So it's looking for these primer sequences, which are, they look to be about 30 base pairs long. We don't want it to find a real sequence and call it a primer. So we're allowing only 2 base pair differences in there. We're allowing it to look not only for the forward read of this sequence, but also the reverse read of this sequence in the palindromics step of it. And then there's another number here, this 10, which says how accurate do you have to be if it's not a full palindrome? And so that means if it's only a fragment of the adapter, how much of it does it have to be? And we're saying it needs to be at least 10 base pairs here. Sliding window, this is what we're just recovering. So we're sliding over 10 base pair windows and looking for the average quality score of that window. And yeah, and so that should generate outputs that we want to work with. Okay, so if we look at LS right now, so we have these outputs. We have R1 paired end, R2 paired end, R1 single end, and R2 single end. And so what we can do here is we can move, we'll do the order on the screen. So we're gonna call up FastQC again, and we're gonna compare our results. So first thing I would implore you to do, going into EcoGeo, into quality control. First thing you want to do is load up this 100,000 R1 so we have something to compare it to. Should go pretty fast. And then we'll open up, and then we're going to say you can go here. So right now, if you look here, you can't actually see, there's nothing called R1.paired end. It's because right now it's only looking for things that have a fast Q at the end of it. So we can change this to all files. And that should allow us to see R1 paired end. We can hit OK. And that will load it up. And now if we compare base quality R1 to the new one, you can see we get a much higher, all of the quality scores now are in the green with a lot less variability. So we've thrown out a lot of the poor sequences as part of this. But now we can do things where we've made the quality better, we might have made some more issues along the way. And so so if you remember or we can actually you don't just have to remember, but we can look at it. So sequence length distribution looked like this before. Everything was exactly 100 base pairs long. And now you have something that looks like this, where there's some trailing edge, right? So most of them are still 100 or above 97 base pairs long. So they only had small little clips on them. But there is this long tail now of things that are smaller in size, going all the way down to the 50 base pair bin down here. And so those are all the sequences that have been kept, and we had to quality control them at some length to make sure that they kept in that information. So we were just using good information.
Why not just throw away the short ones altogether?
So you can absolutely do that. And so it's one of those things of trying to just keep as much data as you can. You can set a much higher length cutoff, right? So if we did 75, we wouldn't actually be throwing off too many base pairs, it's maybe a couple hundred, maybe a couple thousand collectively, right? So you could do 75 and it would be everything from this point up. You could do it from 90 base pairs, it also doesn't see, right? And so this would be one of those situations where you can take a look at your data and say, we can just say at 95, we'll keep almost all the exact same data and we run this at 95%, a 95 base pair cutoff, and we'll keep all the data, right? Just to give you an idea of what you could do with this. All right, So yeah, so if we go back out, and I can give you an example of this. So I'm going to close out of FastQC. If you would like to take a minute and go through and look at the R2s, I promise you they look the same in terms of change in quality score. But you know, if you don't believe me, that's fine. I'm not saying that you're calling me a liar. I'm just saying that maybe just believe me, it's fine. But what we can do here is we can use our handy dandy move command that we learned before, and we actually rename this into something meaningful like I was saying before. So we can do move R1_PE, and we can change this now to be IGM 100 R1 100,000. Here I can add in a trim.fastq. And so remember with move, you're going to delete R1 and _PE, and it's going to be completely just renamed as this new file. So if we do that, we can do that, and we can do the same. So if you hit up and change all the R1s to R2s, you do that as well. And if you hit ls, then we have our data here, and now we've indicated in the file name that we've trimmed it. And so having long file names is not a problem. It is very similar to those of us who do this with our manuscripts when we're writing them, where you have final version 1, final submitted, right? It can start to lose some meaning if you are adding all of the information you've ever done to the file. But having good markers for what you're doing allows you to look at something that maybe you're coming back to in some future time period where you haven't been looking at the data every day for a while. And you can look at something and be like, oh, this is trimmed. I do this when I trim my files. So this is a clean file. I can work with this in a different way than I would if it wasn't a trim file. Okay, so that's Trim-O-Matic. So now we can switch over and try and compare this to using a different set of tools, CutAdapt and Sickle. So CutAdapt cuts adapters from the sequences. It can target an input sequence that has a fragment and locate all possible positions of your adapter. So there's one positive of this. One is that it's tactile. You decide the sequence that you want, and you can decide where it's being searched and what you're doing. And you get a much more feedback than like I put in a FASTA file that had some Illumina sequences in there, and I hope that I was right. The downside to this program is something that gets corrected when we move into the Sickle part of it, is that it doesn't maintain paired end reads. And so you can get a sequence that gets fully trimmed to 0 length. And then because it's not keeping track of that, you end up with this order where like instead of having paired end as one file and single end ones as another file, you end up with something that should be singled end matched up against something that's 0 base pairs long. And Sickle will correct that for us, but CutAdapt by itself doesn't solve that problem. So we can run CutAdapt. And so in this case, we're actually going to write out the sequence that we're looking to remove. So again, you can follow along here on the command line. So it's CutAdapt is the name of the program, -A is for adapter. And then the sequence is AGAT GGAA GAGC. All right. And so right now we're just looking for the adapter sequence. We can add additional flags like the -G. If we only want to look for it at the beginning of the sequence, we know this is the forward adapter. It definitely doesn't appear on the other side of the sequence. We can do a -G, or we can do -B, which looks for the adapter anywhere. So it doesn't just have to be at the forward part of the sequence. It could be at the end. So if there was a mistake in the library and it was being read incorrectly, you can do it that way. We're going to tell it to use an error cutoff of 0.08. So that will give us, that's at about 2 base pair flexibility in the match. We're going to say that it needs to have a minimum overlap of at least 3. So please don't find AGC don't find AGC anywhere in the sequence and delete it. Like that would be a bad idea. We don't want that information to be, that's actually, a real triplet that could exist in the real world. And then our output is going to be a shorthand where we're going to call R1_cut. We're going to tell it what sequence we want to work with, which is the R1.fastq. for the 100,000 one. And then there'll be some information that this generates that we can actually visualize after the fact in a document that we're going to call cut_R1_summary. And so this will give you some statistics about how effective this was in its processing. And if we hit enter, it should run. And then we can look in to the cut R1. And so this is going to give you information about it. So of the 100,000, about 2.5% had adapters still in their sequences, right? So, you know, this was data, and again, I'm emphasizing that this was given, you know, data from 2015, and so companies might do a better job at this now. But this was data that was given to us that we were told was quality controlled, right? And here, there's 2.5% of them still have adapter sequences in the position at the beginning of the sequence where we can identify it, and so that's not great. That's some amount that we don't want to be there anymore. And then it will give you a bunch of information about the base pairs and what's happening there, and then how many times it had to make a cut at a certain length, right? So there were only a handful of sequences that need to be cut anywhere other than the first couple base pairs. But then this is, you know, so 663 had to be cut in their first 3 base pairs because of this, first 4 base pairs, first 5 base pairs, to just give you an idea of the frequency of this happens. So there are some down here where there were the forward primer was identified at the end of the sequence. So for 56 sequences, the forward primer was all the way at the end of the sequence and it had to throw the whole sequence out. And that's just, you know, the luck of the draw. You know, when you're generating 25 million or 100 million sequences from on a sequencing run that you do nowadays, some of them are just failed chemistry. That's going to happen, right? The same way that sometimes when you run a PCR, your PCR doesn't work and you're like, well, I've done it exactly the same for 3 years in a row. What happened? And you start to freak out and you question everything about your life. But the same thing happens in your sequencer runs as well. So some fraction of those chemical reactions fail. You end up with something that looks like data. The computer thinks it's data. It gives you something that it says is data. It turns out not to be data. And so we can screen for that as well. And so I'm asking this question of why are we using this letter sequence for the adapter? And it's because, so if you were to go in and use the NCBI, so this one in the virtual machine is a little out of date, but if you follow this hyperlink, it would take you to the NCBI UNIVAC database, which has over 2,000 fragment sequences for primers, adapters, and cloning vectors. If you've ever tried to submit something to NCBI as a sequence or like a genome, and you have any of these left over, they will yell at you for having them in your submitted genomes. And so this is one of the Illumina primer sequences. You can see here it's highlighted in black. And that's what we're looking to go after and find and remove. And we looked at the less summary. Okay, so one of the things that we can also do using CutAdapt is we can use a FASTA file provided by somebody like NCBI and essentially do the same thing that we did with Trimmomatic, which had like a source file that we could use to compare against. So in this version of it, we're going to redo it just a little bit, where we're going to make it this is a little higher threshold, a little lower threshold in terms of the quality that we need to keep it at. So it's only 2% instead of 8%. The overlap that we're going to check for is 5 bases instead of 3 bases, and we're going to use all of the sequences in the UNIVAC adapter file. So that should be here. So if we hit LS, you can see it's right here. When we run this, we're going to overwrite our R1 cut and our cut R1 summary file, which is okay. This is what we're going to work with. So we're going to do CutAdapt. We're going to spell correctly. -B to tell it that we're going to look using a file, and we're going to use file. Okay, so -B is we're going to look for it anywhere. We're going to tell it it's a file. And then we're going to type file --, and see if we get, and then if you type NC and hit tab, it should autocomplete. So you don't have to write all of that out. And there'll be, so I made it so that because we are looking anywhere in the sequence, and it can be any type of sequence that has been previously identified as cloning vector or a primer of any capacity. Again, because you're in a lab facility where these labs that you're sending sequencing to nowadays generally are doing lots of work. They're not just doing one thing. You can easily get contamination in the air that gets into a couple percentages of your sequences that then aren't real, and we can find those and we can remove them in the same way. And so because we're looking everywhere, I've lowered the threshold just a little bit to give you an idea of what I can look at. We're going to do, so dash E 0.02, overlap equal to 5. And then our output is going to be R1 underscore cut. Same input, IgM 100,000 R1 dot fastq. And again, we're going to put it into our cut R1 summary.
How do you pick your overlap number?
So this is one of those things. So this is a threshold, right? So this tells it how many base pairs it's looking for compared to what the adapter is looking at. How many of those in a row does it have to be to find that? So generally, if you're only looking at one part of the sequence for like the 3' end or the 5' end, because it's looking right there, that can be a pretty short number. So that's why we use 3. So 5 here probably is still maybe a little too short because it can be any 5 base pair alignment of the primer sequences I was looking for. It can be anywhere in the sequence. But this is where you would look at it and be like, okay, so I'm using all possible primers, right? At some point, there's some statistical average where if you're saying the minimum can only be 10 base pairs long, for that to randomly appear in nature, a 10 base pair fragment is like an infinitesimally small scale, right? So it should only occur if it's occurring in an artificial primer or an artificial sequence the couple of times that it's carried over into chemistry. But there is a good chance that you will also find real data, especially if you get smaller. And you can play around with this. So if you wanted to run this a couple times, see what it looks like if you did a 10 here and see what you would do if you would do a 2, right? You would actually see there's probably a pretty substantial difference in when you get it as an output. All right, so if we hit enter, it's gonna take a little bit long because now it's instead of just looking for one sequence like we gave it before, it's looking at the whole data set, which has 2,000 sequences in it. That should run. The B flag means that it looks anywhere in the sequence for it. So now you can do less R1 and the information's there. Less cut R1 summary. You can look here and see how does that compare. So if before we threw out 2.5% of our sequences, here to be a little bit more conservative using all of the possible vector primers that NCBI has, we're now throwing out about 5.2%, which is not that big of a difference, but it can, can be useful in making sure the quality is at the high enough level. And here it'll tell you which of the particular vectors it's identifying, right? And so a lot of these is just a very small number of things that it's identifying, because in theory, we shouldn't be seeing these except for the random times that they occur, right? We should not be seeing whatever this happens to be. This is some vector for some primer or some vector for some plasmid, right? Like it shouldn't be occurring too often in our sequences. And you can see it really only happens in a very low threshold, right? So like if we were looking at this and we were seeing a lot of times, we were seeing 5s, but we weren't seeing 8s, right? This is where we could decide our overlap value a little bit more effectively, because if we set an 8 here, we'd only throw out 2 sequences, whereas opposed to right now where we're throwing out almost 100, right? Just based off of random chance.
So this could happen through contamination, like we said.
Can happen through contamination, or it can happen through actual general
like
occurrence.
errors.
Yeah.
Okay.
Or a real occurrence of the sequence in there. Yeah.
It happens in reality.
Sequence happens.
Yeah.
So yeah, you risk throwing away real information.
Right.
Yeah.
But only about 40 at a time, right? So it's so that you can adjust it. But again, if you were to set the 8 threshold here instead of 5, we'd only be throwing out 2 here. We'd only throw a handful out here. We'd actually keep a lot more of our data in that case too. So this is one of those things that you can play around with and find the right mixture for your data. I actually don't know if there's any that are like, this is definitely contamination in your sequencing. So somewhere in there might be something that looks like that. Okay, so we want to repeat this step, but for R2. So what we can do is we can hit up twice. We can just change all our R1s to R2s. Going back, make sure you change R1 cut to R2 cut. And once you've done that, you can hit enter. And then again, if we check, we can check cut summary R2. So this threw out again about 5.2%. And that gives us a similar type of information. Again, showing that maybe 8 would be a better cutoff for looking for the difference between real sequences and not real sequences. And so, this is something that, the reason that we do this first is because, like I said, no matter what, we do this, there should be some assessment of the quality of your sequences, no matter the type of project you're working on when it comes to a sequencing project, right? Amplicons, metagenomic, transcriptomic. But, a lot has changed in the last couple of years, right? So this is already using an older chemistry of Illumina. There are new chemistries of Illumina. We find in the environmental group, I don't know if everybody agrees with this, that when a new chemistry of Illumina comes out, it somehow happens to have way more quality errors than the last time they had a new chemistry of Illumina come out. And that has to do with the optimization of samples, right? Everybody spends some time figuring out the best way to get a library to work or sequence generation to actually work. And so when something new comes out, the method that you were using previously might not be as good for the new chemistry. And so we found that when you switch over the chemistries, sometimes you end up having this real big drop in for some reason, I'm throwing out half of my sequences this time instead of 20% of my sequences. And so it is good, especially if you're looking at something new, to do this type of assessment. All right, and so now what we have to do, now that we've trimmed away our adapters, is now we have to run Sickle to actually do that quality score that we did in Trigonomatic. We're going to pick a range, a length, and we're going to look for an average quality score and trim based off of that. And so what we can do, this is okay. So right now we have 2 paired end files, right, that have the names in the same order, but in some situations, you can have a paired match where some of them have no length and some of them have a length. And so we're going to actually put both sequences or both forward and reverse reads into Sickle, and Sickle will look for that and remove pairs that have an issue with that. So we're going to be okay. It's not going to be a problem. But you do need to make sure that if you can't just run CutAdapt and then use that output in the next step, because you're going to run into errors where the paired ends aren't matching. So we're just going to type this out. We're going to do Sickle. Again, we're telling it's paired end. We're going to tell them which files we're using, which is R1_cut. And our forward read is the F-R for R2, which is our reverse read. Again, we're telling it it's using, so then instead of saying Fred 33 here, we're saying Sanger. So these are the quality scores based off of the Sanger method, or the Sanger threshold, which is that 0 to 40 score range. We're going to use the same quality score as we used before, which is 28. We're going to use the same minimum length, which is 50. We're going to trim anytime there's an ambiguous base, right? So sometimes the sequencer will put out an N to indicate that it didn't know what base it was. We're going to trim there. That's going to count as a mismatch. And then our output's going to be R1_cut_Sickle. And then another one there. So O is the forward reads, -P is the reverse reads, which is -P R2 cut_Sickle. And then we're going to do -S, and anything that's identified as a single read, right? So the ones where they don't match, we'll call single end cut Sickle. And that should run.
I may have missed this, but why did we choose Sanger over Illumina?
So the Illumina, no, it's not. So this in Sickle, this Illumina is early Illumina,
Okay.
which also uses the Selexa, the Selexa quality score range. I know, it's super useful, right? All right, and so there's some information here. So our paired ends are being looked at. We're looking at a total of 100,000 pairs or 200,000 sequences. And we can see how many we've kept as pairs, how many have been kept as single end, and then how many have been discarded. So in total, we've discarded 43,000 of the sequences, so about 40%. And so we can actually compare the results of this. Or first we can look at the quality. So we can do FastQC and do a one-to-one comparison to all of this. So if we do open, EcoGeo, quality control. So the first one we want to do, right, is we want our R1, and this is our raw data. Should load up pretty quickly. And then we open. This is our trim data using Trim-O-Matic. That should open pretty quickly. And then we can do one more time. If we open, again, changing sequence file to all files, and we look for R1 cut sickle, which looks like this. Hit that. We can compare and see what these all look like. So if we do this, we do that. So here's our raw data. Here's what came out of Trim-O-Matic. Here's what came out of Sickle. So fairly identical. And if you look at here, the values, the difference between it is about 2,000 sequences, right? So it's about 2% of our data. There's a slight difference between it, but it gives you the options of being, like I said, Trim-O-Matic is one of these things where you can put all of your stuff into it and you can do it all at once, and you can get an output. And then, if you use CutAdapt and Sickle, you can break it down and put it into things and get a better idea of what's happening on each step as you go along. And again, there's, so this was again 2015 when the original virtual machine was made. These were the programs I was using. There are probably another 150 programs that have come out that will trim your data the best way possible with the fastest counts, using all sorts of fancy new technology to do it faster and better. And you're free to use those. But this is an important step, and it's something that you should do to assess the quality of your reads before you go any further. I had a hand, yeah.
How do you know what quality score type is being used from any particular sequence?
Yeah, so the question is how do you know what type of quality score is being used? So the rule of thumb right now for this is really Illumina is using the original Fred-based quality scores, which is 0 to 40. And then the newer, longer reads, I actually don't know what quality score systems they're using. But in general, it was 454, Selexa, Ion Torrent. They all had different read quality score values, but they've really fallen out of favor on the grand scheme of things. And they're not really they're not really using that in the same way. So the real thing is like if you're using an Illumina sequencing platform, it's using the quality scores that have been around for a while, that 0 to 40 range. Any other questions? Yeah.
So you talked about newer softwares that have come out.
Yeah.
Have you ever used FastX Toolkit?
I've heard of it. I have not used it, no. Yeah. And again, so this was a very conservative way of doing this. So I know there are definitely, I've given this flow through to people, like workflow to people before. And they've been like, I just threw out 35% of my data. And I'm like, yeah, but you only have the good stuff left. And then they'll go, I don't like that. And then they'll go somewhere else and they'll find something that might only throw out 15% of their data or 10% of their data. And their curve will look different. You can go in and you can visualize how that works. It's how much you want to give it, right? What was it? I've just read a paper the other day where their average wasn't 28, their average was 20. And they were happy with that. And that was fine with what they were doing. And it means that their curve dips a little bit lower as the length of the sequence gets longer, but it also means that they keep more of their data. And so it really depends on what you're trying to do. I can easily make a case up here that if what you're trying to look for are genuine variants, like in the environment, using some type, transcriptomic, where you're going to map it onto something, you don't want to have something that has a quality score of a 20 in your reads, right? That's just not what you want to do. So I can look back here when you're doing lots of whole genome, and you're trying to map reads back to your starting genome, you want a really high quality score, right? You don't want something that's coming in as an error that, if you get enough reads, becomes a statistical error that might look real but isn't real. But at the same time, if you're doing something where you want to capture as much data as you can and you have an assembler that you're confident will work with it, you can go the other way and be like, yeah, we're going to use an error 20 as our cutoff. But yeah.
We have two questions. You mentioned we should remove primers.
Yeah.
Did we remove primers?
Removing the primers? Did we remove primary? How did we? Is that the question? So in Trimmomatic, there was a file that kept it stored for us. And then when we moved to CutAdapt, we actually gave it the sequence we were looking for. And then also there was a file that had the primers in it.
Isn't that
So it's adapters and primer sequences in the NCBI setup.
second? We have this blog that we don't really need to remove the primers because it's natural part of the cell.
Sure. So if you're doing something for like amplicon sequencing, right, you can keep the primer, right? It's just a natural part of the sequence. For metagenomics, right, the only primer that's in there is the sequencing primer, which is not a natural part, right? So it depends on the method that you're working with. And that's not a raise a hand. You don't have a comment there? I thought Kristen was gonna. I thought you were raising your hand to be like, but I have a. Yeah, but so that's for like amplicon sequencing, right, where the primer is actually in the sequence. That's what you're targeting. For metagenomics, there's no target, right? So you don't want to find primers in there, even by accident. Any more questions? So that was a nice, perfect hour-long session. There are some drinks in the back, sodas and stuff, if that's your fancy. I think at some point in the next half an hour or so, we should get a delivery of snacks for the afternoon. That should come in. Yeah, I know. And if they don't, I'm going to have to find out why, because we're paying for it. And so that's it for right now. We're going to switch over. Kristen's going to take over for the Chime 2. Everybody can get up and take a break because I need a minute to stop the YouTube live stream and put on a new live stream there. Ooh, shoot.
See if they could move your break up.